Well, good morning, good afternoon, or good evening, ladies and gentlemen. Welcome to another edition of our video PowerPoint series for Unit 5.5, Mitosis and Meiosis. So, there's not really enough in this unit to call it its own unit, so it's kind of a 5.5, an intermediate point, if you will. So today what we're going to be talking about is the cell cycle and mitosis. As you can see here, there's no real video in the upper right-hand corner, so unfortunately, or maybe fortunately for you, depending on how you feel, you will not be seeing my wonderful face here. This will be an audio only. So to go ahead and kind of counterbalance that, here's Macho Man Randy Savage. Just soak it in for a second. Alright, anyway, so today we're going to be talking about mitosis and meiosis. So, go ahead, get your notes ready, and listen to the sultry sound of my voice as we work our way through mitosis. Let's get started. So, the big question here is, how do little elephants grow up to be big elephants? Now, we know for a fact that the size of the cell isn't increasing, because we know that gigantic cells are pretty terrible when it comes to carrying nutrients. So what actually happens is, it's not the size of the cell, but it's how many cells the organism has. The more cells, the bigger it gets. Speaking of that, why do you think animals shed their skin? Well, simply put, imagine it like this. As you grow, you always get new socks, right? Why? Because your body is growing. Same thing with animals, specifically reptiles and crustaceans. What they need is, instead of replacing their sock, they actually have to replace their skin. So when you see an animal molting or shedding its skin, that's basically what's going on. Fun fact, we actually do shed our skin, but not like we can't just peel it all off. Our skin cells are actually dying and regenerating all the time. The more you know. So the process of asexual reproduction begins after the sperm fertilizes an egg. So if you remember, asexual is one parent, right? Whereas sexual is the combination of two sets of genes. So, it's simply put, asexual reproduction begins after the sperm fertilizes the egg or after sexual reproduction occurs. So there are three reasons why cells reproduce by asexual reproduction. The first is growth. The second is repair. And the third is replacement. So as we grow, we need more cells. And when our body gets damaged, we have to repair those cells. And also, like we said before with the reptiles, we have to replace cells that are either damaged or destroyed. For example, skin cancer is the abnormal growth of skin cells. Most often develops on the skin due to exposure to the sun. Cells that reproduce by asexual reproduction are reproducing constantly. So right now, you've got cells that are reproducing inside your body as we speak. So cells multiply by dividing. In eukaryotes, we go through the process of mitosis and meiosis. In prokaryotes, they go through the process of prokaryotic or binary fission. Now, we talked about binary fission before. That's basically just the cell splits itself in half. Mitosis, however, the main distinction between the two, mitosis and binary fission, it's true both of them split the cells, but mitosis involves nuclear division. So we're dividing the nucleus, which is something that only eukaryotic cells have. Now, inside the nucleus, we have these things called chromosomes. Now, chromosomes are actually what contain the genetic information, or DNA, that is passed from parent to offspring. They look kind of like this. So imagine a really fuzzy X. That's pretty much a chromosome. So chromosomes have two parts. They have the chromatids, which are held together by a centromere. So if you wouldn't mind going ahead and sketching this in your notebook. The centromere is the central part, where the two sister chromatids are held together. Meanwhile, the chromatids can be found here. So we have 1, and we have 2. Two different chromatids held together by a centromere. The chromatids are exact copies of each other and are split apart during mitosis to form single chromosomes. So, as we go through mitosis, we're actually going to go ahead and split these two apart so each cell gets an identical copy of the genes that it needs. So, chromosomal replication. We go from one chromatid, and when we reproduce, we go to two sister chromatids. So, a sister chromatid, well, we call sisters pairs of chromatids like this. So anytime we go through replication, we're going to go from one chromatid to two sister chromatids. When cells divide, sister chromatids separate so that each cell has the correct number of chromosomes. So as we can see here at the top, here at number 1, the chromosome is unduplicated at this point during interphase. So once we go through a certain part of interphase, which we'll talk about in a bit, the chromosome will duplicate, making an exact copy of each other. Well, not each other, of itself. Now, once mitosis occurs, we're gradually going to go ahead and split these two apart, so that each cell gets a copy of the original genetic material, making it identical. So the main question here is diploid or haploid. First off, what the heck does that even mean? I mean, you just threw two words at me. How am I supposed to know that? Well, hang tight, because I'm going to explain it. A diploid means 23 homologous pairs. It's the full number. So in humans, the diploid condition is 46. 46 chromosomes in each of your cells. We call these somatic, or body cells. These are every cell in your body except for the sperm and the egg. On the other hand here, haploid cells only have 23 chromosomes, or half the full number. These are the gametes, or the sex cells of the sperm and the egg. So think about it. Why do you suppose that the sex cells, the sperm in the males, and the eggs in the females, only have 23 chromosomes? I'll let you go ahead and think about this. Keep it in the back of your head. If you know it, go ahead and keep it to yourself, because unfortunately this is a recording and, well, I can't hear you. But I'm glad you thought of that anyway. Good work. I'll see you on the next slide. So in humans, human diploid cells have 46 chromosomes, or 2 copies of 23 different chromosomes. And the chromosomes here you can see to the right. So we have 22 pairs of chromosomes, and then we have an X and Y. So we have an X chromosome and a Y chromosome. Now, we'll talk about this later, but the X and Y chromosome more or less determine the gender of the offspring. If you have an X and a Y, that's going to result in a male. Meanwhile, if you have an X and an X, that is going to result in a female. I understand that those are two terrible drawings, but I'm using a mouse here. I'm doing the best I can. So the cell cycle for somatic cells, remember somatic meaning body cells, has two main parts. The first is interphase. This is the life or growth phase. The second is mitosis, which is the division of the nucleus. Now we also have cytokinesis. However, for the course of this class, we're going to consider cytokinesis part of mitosis. So let's go ahead and talk about these cycles individually. With interphase, interphase is the longer part of the cell cycle. The cell roughly spends 90% of its time in interphase. Interphase is composed of 3 different phases. The G1, or the gap phase, the S, or synthesis phase, and the G2, or the growth phase. So, once again, we'll talk about these specifically coming up. During interphase, the DNA is relaxed and spread out thinly. So as you can see here, you can't really see the chromosomes yet. Everything's kind of all bunched together inside of our nucleus. And the nucleus is still together. This is when the chromatin, or chromosomes, are thin, stringy, and invisible. The chromosomes are what we call chromatin. So before we actually have the chromosomes, the two X-like DNA storage molecules, we call them chromatin. So the three phases of interphase. We have the G1, or the gap phase. This is when the cell is living and doing normal cell processes. The S phase, or synthesis phase, is step 2. This is where the DNA goes ahead and replicates. So now we have a copy of the genetic material inside of the cell. During the G2, the cell is expanding and replicating its organelles, getting ready for division. So by the time we're done with the S and G2 phase, basically what we have is a cell that has double all the organelles and double the genetic material. We do this to make sure that each cell created during mitosis has the correct number of organelles, DNA, and well, everything else that it needs to go ahead and survive. As soon as the S phase starts, there's no turning back. The cell is without a doubt going to go ahead and divide. So the big question here is what happens after interphase? What happens during that other 10% of the cell's life cycle? That's what we call mitosis. So cell division. Mitosis, if you remember, is what we call nuclear division. Cytokinesis is cytoplasmic division. So we're going to call both of these together the process of mitosis. So think back. Why exactly do cells divide? Take your time. Well, there's 3 reasons, remember. We have growth, repair, and reproduction. Growth and repair are primarily handled by mitosis, whereas meiosis, which we'll talk about in the next video, is responsible for the reproduction. So in mitosis, diploid, remember 46 chromosomes, somatic cells, or 46 chromosomed body cells, divide to become diploid somatic daughter cells. Remember, mitosis makes exact copies. So, if the if the cell had 46 chromosomes to begin with, when it makes an identical copy, you better believe that that also is going to have 46 chromosomes. So, mitosis making an exact copy. So, mitosis has 4 primary phases, plus cytokinesis at the end. We have prophase, metaphase, anaphase, and telophase, which we're tacking on with cytokinesis. So those are our 4 steps. As we progress here, we'll talk about each one individually. Now this can be found on page 246 in your workbook. Or if you can't find it on there, it should be, well, in the ballpark. Give or take a couple of pages. So before we go any further, let's learn some helpful hand signs. Now you can go ahead and do this yourself. I'm going to go ahead and assume that you're doing it, or at least looking at the screen like, dude, there's no way I'm going to go ahead and do that. That's lame. But I'm telling you, if you do this, I've seen people do this on tests, and it does work. So we have 4 different hand signs here, each one corresponding to a different stage of mitosis. So we have one where our hands are clasped together, one where they're kind of straight up and down, one where we have 2 almost dinosaur-looking things facing each other, and then we have 2 fists. The first is prophase. The second is metaphase. The third is anaphase. And the fourth is telophase. So, what we're going to be talking about next is what happens in prophase, metaphase, anaphase, and telophase. We call that PMAT. Yeah, that's right. So, on the next couple of slides, we're going to explain what happens during PMAT. Let's get started. Now, you don't actually have to make this table. This table can be found inside your workbook, once again. So instead of going ahead and writing this down on note paper, now it's not mandatory, but you can, there is a table inside your workbook that you might want to go ahead and fill in instead as we progress. So during the first stage here, prophase, the chromatin thickens into chromosomes. The nuclear envelope, or the nucleus, the barrier, disintegrates, and the centrioles migrate to opposite poles. These are our centrioles here. They're going to go ahead and play a pretty significant role in going ahead and splitting our cell. So as you can see, inside the cell here, that chromatin has finally thickened into our X-shaped chromosomes. The nuclear envelope is starting to disintegrate. So as you can see, it's not one continuous circle now. And as we said before, the centrioles, which we have right here, are starting to migrate to opposite ends of the cell. Either here and here, or it could be here and here, depending on how the cell's feeling that day. The centrioles are also going to start producing spindle fibers. These are tiny little web-like projections that are going to be used to pull our chromosomes apart, which we'll get to in a bit. So prophase, why we go ahead and put our hands together like that, is it's showing you that the chromatin is thickening into chromosomes. Our second stage here is metaphase. During metaphase, remember those spindle fibers? Well, these spindle fibers are actually made of something called microtubules. And the spindle fibers are going to attach to the centromere. Remember, the centromere is the center part of our chromosomes. The spindle fibers are going to attach to the centromere of our duplicated chromosomes. So, metaphase, basically what happens is the chromosomes line up along the metaphase plate, or the middle. So why we go ahead and put our hands together like that, is it's showing the chromosomes lining up on the middle of the cell. During anaphase, our third step, the sister chromatids are pulled apart to opposite poles. So, with our hands like this, kind of like you're holding I guess you could say a burger, the sister chromatids are pulled apart to opposite poles. Your hands actually look very similar to what's going on during anaphase. The sister chromatids are pulled apart, and they're going to continue to be pulled apart until they reach the opposite ends of the poles, or the opposite ends of the cell. Finally, we have telophase and cytokinesis. So during telophase, our nucleus is going to go ahead and reform. So the nucleus is going to come back. And the chromosomes are going to relax back into chromatin and go back to the G1 phase of interphase. And eventually, what we're going to have here is cytoplasm. Cytoplasm will split the two daughter cells right down the middle until we have two identical copies of the original cell. So the reason we go ahead and turn our hands into two fists here is it's showing you that the nucleus reforms and now we have two separate cells. We went from 1 to 2. So cytokinesis in animal cells and plant cells, they're a little bit different. In animal cells, basically what happens is we get something called a cleavage furrow. So imagine tying a piece of rope or string around, let's say, a piece like a circle or sphere of jello. If you go ahead and wrap that around there and you keep pulling on that rope, you're eventually going to split them in half. Now this occurs because there is no cell wall. Meanwhile, in plant cells, we can't go ahead and really split it right down the middle because of the cell wall. What we have here instead is something called the cell plate. The cell plate is eventually going to form our cell wall between the original cell, making it go from 1 to 2. Now, as you can see here, this is a tiny embryo. And the embryo is, well, there's something happening to it. So what do you think would happen if cytokinesis failed? So if the cytoplasm of cells never split? Well, we'd actually get something that looks like this. Syndactyl. So what that means is the skin between the fingers, if you look closely, the skin is formed, everything there is fine, but the skin hasn't separated. So there's kind of like a web of skin holding everything together. Now I know if you look between your fingers, you will see little webs of skin there, but I'm talking this skin would be attached up to about your second knuckle. Pretty crazy, right? So, think about what we talked about today. Can you identify the phases of mitosis? And also, interphase? I'll give you a little bit of time here. Remember, you can always pause the video. Speaking of that, I know we kind of went a little quick through all this, but Like I said, you have the benefit of pausing the video whenever you need to. I'm not going anywhere. So, first off, let's try to find interphase. Now remember, interphase, the chromatin's going to be there, nothing is condensed yet, and we still have a nucleus. So if you look at this one up here, in the upper left, that would be our interphase. Now let's try to find prophase. So prophase, remember, the chromatin is going to condense. So now you're going to see these really dark lines, and also there's not going to be a clear ring around the nucleus. This one in the middle will be the closest thing we have to prophase. So let's see if we can find metaphase. Remember, metaphase is where everything aligns on the middle. So the chromatin, or at this point their chromosomes, are lined up along the middle of the cell, which looks a lot like what we have here. Our next step is anaphase. So anaphase, look for the one that looks like the chromatin, or the chromatids at this point, are being pulled apart. Bingo! That would be number 4. So by process of elimination, we know that this must be our telophase. So telophase, as you can see here, the chromosomes, or chromatids, are finally starting to go ahead and recondense back into our chromatin. The nucleus is gradually starting to reform, and as you can see, the cell is being pulled apart into 2 different cells. So those would without a doubt be our 5 stages of mitosis, including interphase. So if you look here, you can actually see the same thing. So this would be our interphase, our prophase, our metaphase, our anaphase. I wanted to write the letter A there. Let's turn that into a 4. And our telophase and cytokinesis. Now this is actually what it looks like under a microscope. So if you ever take a look at real cells while they're dividing, it looks similar to this. So a quick summary here before we go ahead and break. What we have for mitosis is we have primarily mitosis and interphase. So during interphase, we have the G1, which is the first growth phase. It's primarily where the cell does its normal cellular activities. We have the S phase, or synthesis phase, where the DNA goes ahead and replicates. So we have twice the amount of genetic material now. Our next step is the G2. The G2 is where we go ahead and duplicate all of our organelles and prepare to divide. From there, we finally go into mitosis. So we have prophase, metaphase, anaphase, telophase, and finally cytokinesis, finishing off mitosis and the cell cycle. So, ladies and gentlemen, that's just about going to go ahead and finish this up for today. So today, in summary, what we talked about, well, actually, you know what, we don't really need to do this. We talked about the summary in the last slide, but basically we talked about what mitosis is, the division of body cells with 46 chromosomes, and making identical copies of them. Now remember, mitosis is nuclear division. So for it to be called mitosis, there has to be a nucleus, which is why prokaryotic cells go through binary fission. So we talked about what interphase was, what mitosis was, and the steps of each. So that's just about going to go ahead and finish this up for today. So ladies and gentlemen, thank you very much for joining us today. And until next time, I will see you in the next video. You all keep it classy.